In this video we will discuss about glycosylation in gliomas. So, and the disruption in this glycosylation in gliomas can lead to the uncontrolled proliferations, and that could be the characteristics of the cancer cells, and these are usually found in the brain. So we will start with this regulating protein functions. So how this glycosylation in these gliomas regulates the protein function. So there are two different categories. First one is the direct effect, that is the proper glycosylation will helps the proteins to reach to their final destination state. Uh, for examples, these proteins are expressed. These are the glycosylated proteins that are expressed on the cell surface. And the indirect glycosylation is that the proteins that are decorated with the carbohydrates or the glycans will get disrupted if the enzymes that are responsible for glycosylation are not properly expressed within the cells. So this is not just specific to the glioma, however it is more common to all the types of cells. And now how this glycosylation will affect the cell-cell matrix or cell-cell interactions. In the case of glioma, what people have observed is that the upregulation of this GRP56 will affect upregulation of this GRP56, and this truncated PTP new formations will affect the cell-cell interactions. And also since this uh, alpha beta integrins or these integrins are also glycosylated, so disruptions in this glycosylation will also affect the functions or the contributions by these integrins towards the cell-cell interactions. So here the three different kind of disruptions that might cause or that might affect the cell-cell interactions, that is upregulation of this GRP56, truncated PTP new, and there's dysregulations of the glycosylation on this integrins. So thereafter because of this change in the glycosylation, how it will trigger the downstream signaling pathways. That is, if this phthalate which is responsible for glycosylation get disrupted, what it will do is it will affect, first it will affect this MET, that is mesochemicals to epithelial transitions. Uh, for example, it will upregulate there are high mesochemicals to epithelial transitions, and when this LCA interact with these receptors, HGF or MET, and the signaling pathways will cause proliferations, invasions, and migrations. And similarly, for this EGFR receptors, it will trigger high signaling pathways. But these EGFR receptors not only depend on phthalate, but it will also depend on GALN-T2. If there is upregulation of GALN-T2, then it will trigger upregulate the survival pathways, and if it is downregulation of GALN-T2, then it will suppress the survival pathways. And about this GRP56, what will happen when these GRP56 are overexpressed, then it will form a will helps in the formation of this beta catenin and TCF in the cytosols, and thereafter it will also helps in these invasions or migrations. And these integrins, about the integrins, if this beta 4 GALT5 are formed within the cells, then it can interact with these integrins and helps in this phosphorylation of this ERK1, JNK1, AKT1 pathways, and that helps in uh, this migrations or MET transitions of the cells. And however, in order to activate these integrins, it needs to have a sufficient amount of this beta 4 GALT5 formations. And as we know that beta 4 GALT5 formations, in order to form a native structure of this beta 4 GALT5, it needs to pass through a quality control mechanism within the endoplasmic reticulum. And if there is a less population of this HSC70, then the quality control checking of this or this HSC70 helps in this native structure formation of this beta GALT T5. And there is a low amount of the HSC70, there is less native formation of this beta 4 gal T5, and because of that the integrins will not be able to perform its normal functions, and that will lead to this phosphorylation, this uh, downstream signaling pathways. So this one of the problems that observed in this gliomas is that HSC70's chaperones formations are low, and because of all this combined effect, what it will cause is it will cause the malignant progression of these gliomas. Uh, that is, it is not only have the uncontrolled growth rate, but it will also spread from its origin to the secondary organ sites. I hope you got a basic idea about this glycosylation in gliomas, and what are some of the changes that might cause, or that might cause these gliomas to have this proliferation, invasion, and migration characteristics. If you need to understand more in detail about this changes in the glycosylation in gliomas, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.